This meeting is being recorded.
In this video, we'll be seeing about biomolecules. Biomolecules are the chemical compounds that are found in living organisms. They include both the organic and the inorganic compounds, which means they contain carbon, that is, compounds containing carbon, and there are also compounds that do not contain carbon. How do we analyze the chemical composition of a tissue? So, we take any tissue. We want to find what is that tissue composed of, what are the basic chemicals that is present inside that tissue. The first step is we will grind the tissue using a mortar and a pestle, as you can see in this picture. And this living tissue can be a vegetable or a piece of liver, anything. And you will be grinding this with the help of trichloroacetic acid. And when you completely grind it with the help of trichloroacetic acid, you get a slurry, and a slurry is this. You'll be getting a substance like this. When we strain this, that is filter this through a cheesecloth, which I'm marking it here, which is the one which is used for making cheese, or cotton, we will get two fractions. One is the filtrate and the other one is the retentate. So what does that mean? One of the filtrate will be soluble in acid, other will be acid insoluble. And using this method, they are named into two different categories. One is the filtrate, other one is the retentate. Filtrate contains the biomicromolecules, which means their molecular weight is less than 1000 Daltons. The retentate contains the acid insoluble fractions. So they contain the biomacromolecules, which have the molecular weight greater than 1000 Daltons. And quick recap here, Dalton is the unit that we use to measure the atomic level. So how do we analyze the inorganic compounds? We can weigh the living tissue in the normal condition, which is called as the wet weight. So it will be hydrated, it will have some moisture, and it will have some water content in it. And followed by that, it is dried, and then it is weighed again, which will give the dry weight, and which will not have any water, as the water would have evaporated. Next, it is fully burned to oxidize all the carbon compounds. So when you burn, obviously oxidation happens. So all the carbon compounds gets oxidized and they turn into gaseous form, that is carbon dioxide and water vapor, and finally it forms ash. This ash which we get from here will contain all the inorganic elements like calcium, magnesium, sodium, potassium, and the inorganic compounds like the sulfur dioxide ions or the phosphorus ions, sodium chloride and calcium carbonate, etc. This is an interesting data that gives the comparison of elements in both the non-living and the living matter. So the basic elements such as hydrogen, carbon, oxygen and so on, what is the total percentage of that present in the Earth's crust, and what is the comparative amount of the same present in the human body? This slide gives a complete picture of both. I'll pause here for 10 seconds so that we view this slide. Going to the next one, the biomicromolecules. The molecular weight of biomicromolecules, as said earlier, ranges from 18 to 800 Dalton. That is, they are less than 1000 Dalton. And they are also called as micromolecules or simply biomolecules. And they will be found in the acid soluble pool. And this acid soluble pool basically represents all the cytoplasmic compositions, which means all the components, the organelles or the chemicals that are basically present in the cytoplasm of a cell are the biomicromolecules. So the biomicromolecules, or simply the micromolecules, include amino acids, lipids, sugars, and nitrogen bases. We'll start with amino acids. These amino acids are the compounds formed of an amino group, that is an NH2, and an acid group, that is a carboxylic acid, C double bond O, bond OH, and H, which you can find here on the top, and a variable group. So this is an R group, a variable chain. NH2 and COOH, that is the amine group, and the carboxylic acid group, are attached to the same carbon atom. That is, there is a single carbon atom, and it is called as an alpha carbon. Since both of the amine group and the carboxylic acid group were attached to one carbon, and they decide what this carbon has to be changed or substituted into a molecule, and hence they are called as the alpha carbons. And since carboxylic acid and the amino acid is attached to this alpha carbon, all amino acids are also called as alpha amino acids. Apart from this, they are also called as substituted methanes. The reason is, you can find this basically represents a structure like this. So it is basically CH4, and it is CH4 which has been substituted by different kinds of functional groups or different hydrogen bonds. The first one, amine, is substituted here, and then carboxylic acid is substituted here, and there is a variable R chain that is substituted here, and you can find overall that only one of the hydrogen is completely left free. So as a result, you can call them as substituted methanes. So I said there is going to be one change in the R group, and based upon this change in the R group, the type of amino acids is also going to change. For example, if that R is going to be substituted by H, then it becomes glycine. If it is going to be substituted by CH3, then it is alanine. If it is CH2OH or CH3O, then it is serine. You can find in all these three different types of amino acids, the carboxylic acid group and the amine group are not changed. And so does the fixed hydrogen at one corner, and so is the alpha carbon. You don't find any structural differences between them or in the position also. So in anything as well, you will not find any difference except the R group, which makes the big change in the amino acid. There are totally 20 types of amino acids that are used in protein synthesis, and the amino acids are of three types. The first one is acidic amino acids. Glutamic acid is one example. Basic amino acids, lysine is one example, and neutral amino acids, valine is one example. And it is very important to know that some of the amino acids are aromatic, which means they smell good and they are capable of spreading the order of their composition. And as the name suggests, acidic, basic, and neutral is completely on the basis of two factors: hydrogen ion concentration and their reactivity towards an acid or a base. And examples of aromatic amino acids include tyrosine, phenylalanine, and tryptophan. Amino acids are classified into two major categories. One is essential amino acids, the second one is non-essential amino acids. Essential amino acids are those that cannot be synthesized by the body and hence should be supplied through the diet. So they are taken from outside the body as the body cannot produce. Examples of essential amino acids include lysine, leucine, isoleucine, tryptophan, etc. Non-essential amino acids can be synthesized by the body. Examples include glycine, alanine, serine, arginine, etc. So they are synthesized by the body, but they do not find any active usage inside the body. In amino acids, as discussed earlier, the amine group and the carboxylic acid group have an ionizable nature. So what do I mean by ionizable? They can be easily ionized. So when you put them under an hydrated condition, they are capable of changing into ions. So the structure of an amino acid will be changing in different solutions with respect to different pH. And if in any of the solution, and both of them do not get ionized at the same time, they'll be having different pH sustainability, susceptibility, and they'll be changing the different kinds of solutions. But if both the amine group and the carboxylic acid group are ionized, then that is called as a Zwitter ion, where you can find Here the amine group getting ionized, and so does the carboxylic acid group. Next, we'll move to the next biomicromolecule, which is called lipids. In simple, lipids are substances which are insoluble in water and they appear like oil. So, fats. So you can tell they contain carbon, hydrogen, and oxygen, just like a carbohydrate, but the number of oxygen atoms is comparatively less. The lipids are classified into three categories: simple lipids, compound lipids, and derived lipids. A simple lipid is formed from a fatty acid and glycerol. I'm sorry, and an alcohol, and they together form a compound called glycerol. So a fatty acid and an alcohol together will form a simple fat lipid. Fatty acids are the lipids with hydrocarbon chain. So they'll be having an hydrocarbon chain, that is CH, and finally they will end with COOH. For example, palmitic acid is a fatty acid, and this fatty acid will have a lot of hydrocarbon chain. So there are a total of 16 carbons, and they'll be having a very big hydrocarbon chain, and in the end they will be having one COOH group, that is a carboxylic acid group. So their formula will be CH3 CH214 COOH or C15H31 COOH. And there is one more acid which is called as the arachidonic acid, and that will have 20 carbons. And this is the structural, the isomeric structure of such a arachidonic acid. Next, the simple lipids can be classified into saturated fatty acids and unsaturated fatty acids. So this is very similar to the ones which you would have studied in grade 10. That is, the saturated fatty acids or those which will not have double or triple bond, while the unsaturated fatty acids will have one or more double bonds or triple bonds. So example again, if you take for saturated fatty acid, it is going to be palmitic acid and stearic acid. And for unsaturated fatty acid, it is going to be arachidonic acid and oleic acid. Now let us take the structure of a glycerol. If we take the structure of a glycerol, it is going to be CH2OH and CH2OH on the top and the bottom, and supported by CHOH. This is the most simple lipid that we can find, which is called as the glycerol. This is also called as trihydroxypropane because there are a total of 3 CH3. So when you tell C3, basically it represents the propane of the alkane hydrocarbon chain. So it becomes C3. So it is trihydroxy. And since it is hydroxy, because it is substituted by hydroxide on all the three ends, so it becomes trihydroxypropane. Next, the fatty acids can be esterified with glycerol through a ester bond to ester bond. Now let us break this statement and read it. What is the meaning of ester? An ester means they are capable of forming a ketone group. So they can be esterified with the glycerol by the formation of an ester bond. And as a result, they'll be forming monoglyceride, diglyceride, triglycerides, and so on. So how does this happen? We know the structure of a basic glyceride molecule. So we know that this is the basic glyceride molecule. To this basic glyceride molecule, there is one glycerol that is added. And as a result of adding one glycerol with the help of ester bond, it becomes a diglyceride. And if you add one more glycerol to it, it becomes a triglyceride. So one glycerol plus one fatty acid becomes a monoglyceride. One glycerol plus two fatty acids will give a diglyceride. And one glycerol with three fatty acids will give a triglyceride. So the triglycerides we'll take and we'll try to analyze the structure. In this you can find that, let us take this structure so that it will be easy for us to understand. The trihydroxypropane has not been disturbed at all, but instead you can find one fatty acid added here, other one here, and the other one here. So this is what is represented as R1, R2, and R3. So R1, R2, and R3 are the fatty acids, and they form the triglycerides as a total molecule. The second category is compound lipids. These compound lipids are the esters of fatty acids and alcohol, but along with that there will be some additional groups also. So examples. So as said earlier, they will have fatty acids, they will have glycerol, and along with that they will have phosphate, and hence the name phospholipids. They are found in all the cell membranes basically. Lecithin is one example of such molecule. Third one is derived lipids. These derived lipids are not naturally occurring, but instead they are obtained from simple lipids by the process of hydrolysis, which means when we use water, the simple lipids are broken down and finally they will be changing into compounds that is called as derived lipids. So the derived lipids are the ones which were obtained as the products of simple lipids and compound lipids as well. One such example is cholesterol. So we can get to know that cholesterol is something which is produced in our body and it cannot be substituted from outside. The third biomolecule which we'll be seeing is sugar, that is the carbohydrates. The sugars are sweet and water soluble carbohydrates. So they are soluble in water and they are formed of carbon, hydrogen, and oxygen in the ratio of 1 is to 2 is to 1. And so does the formula C6H12O6, C5H10O5, C11H22O11, and so on. So examples of sugar include glucose, ribose, galactose, maltose, fructose, etc. The fourth one is the nitrogen bases. These are nitrogen containing cyclic compounds that are found in the nucleic acids. So basically they are classified into two categories. You can find a lot of nitrogen that is found in the picture that is given here, and it is also very circular. So you can find that it is a circular cyclic structure. Not exactly circular, I'm sorry, but a cyclic structure. The nitrogen bases are basically classified into two categories, the purines and the pyrimidines. So the purines include adenine, guanine, and the cytosine, and the pyrimidines include the thymine and the uracil. The nitrogenous bases form structures called as nucleosides. So when a nitrogen base is added along with a sugar, then you call it as a nucleoside. Here you can find the basic adenosine. That means there is a sugar molecule, and that sugar molecule when is added along with adenine, that is a nitrogen base added with a sugar molecule, it becomes adenosine. So a nitrogen base plus a sugar is a nucleoside. So here you can find that, I'm sorry, going to the previous slide, there is one nitrogen base, and to this nitrogen base there is a sugar base, that is a sugar molecule that is added, and it becomes a nucleoside. So to this complex, if I am going to add a phosphate molecule, then it becomes a nucleotide. So the difference between a nucleotide and a nucleoside is both are going to have nitrogen base and sugar. But the only difference is a nucleotide will be having a phosphate molecule and attachment, whereas a nucleoside will not be having a phosphate molecule. So nucleotides are basically heterocyclic compounds. So what do you mean by heterocyclic compounds? They are cyclic compounds, but one cycle to the next cycle will not be parallel, nor will not be a rotatory in the same direction. That is called as an heterocyclic compound. The nucleic acids are basically the nucleotides, that is the DNA and the RNA. In the next video, we'll see about biomacromolecules. Now we'll continue with biomacromolecules. Biomacromolecules or the biomolecules are the ones which have a molecular weight greater than 1000 Dalton. So they include the proteins, polysaccharides, and the nucleic acids. And their molecular weight sometimes is even 10,000 Dalton and above. And they form a basic composition of the acid insoluble fraction. And they include all the macromolecules from the cytoplasm and the other organelles as well. Now let us see the structure of a cell membrane. When you see the structure of a cell membrane, it consists of a lipid bilayer which is getting segmented here and there by a transport protein and some phospholipids sandwiched in the middle and some proteins floating on the top. And these proteins are basically attached to the carbohydrate chains. This is how the structure of a cell membrane is. But if you see lipid, it is not a macromolecule at all because its weight does not exceed even 800 Dalton. But it comes under acid insoluble fraction because many lipids are arranged into structures like cell membrane. So the cell membrane it appears a mass structure like a cell membrane. So when a tissue is basically grinded, the cell membranes are completely broken and they form water insoluble vesicles. So vesicles means globe-like structures or small fat-like globules which will appear bigger. So they cannot be filtered along with the acid soluble fraction. As a result, you call them as a macromolecule. The average composition of a single cell will be in this way. Water will be 70 to 90%. The protein will be for 10 to 15%. Carbohydrate is just 3%. Nucleic acid is 5 to 7%. And ions will be forming just 1% of the total composition of a cell. Going to proteins. The proteins are considered to be the heteropolymer chain of amino acids. So basically there is one amino acid and there will be chain of amino acids which is called as a polymer. An heteropolymer means not a same kind of amino acid is going to form a chain. Instead, there are going to be a lot of varieties of amino acids that is going to form chains with one another, and that's what you call as heteropolymer chain. And they are polypeptides, that is linear chains of amino acids, that is poly, and peptide, they are linked by peptide bonds, and hence the name they are polypeptides. And when does this polypeptide bond form? A polypeptide bond is formed, a peptide bond is formed when the carboxyl group of one amino acid reacts with the amine group of the next amino acid by releasing a molecule of water in a process called dehydration. So you can find how this happens. There is a CO group here, COOH, and it bonds to the amine group of this amino acid. Let us name this as 2 and let us name this as 1. And as a result, water is released, and this water which is released in the form of dehydration is called as the wastewater, and in the process it forms a bond which is called as the peptide bond. So here you can find A1 is an amino acid, A2 is an amino acid, A3 is an amino acid, and so on. And all of these are linked together with the help of peptide bond in the middle, which is represented by the white line. So this chain of amino acids together is what we call as the protein. What are the basic functions of protein? The proteins are used for growth and tissue repair. They transport nutrients across the cell membranes. They act as intracellular ground substance. We'll see about that little later. They act as antibodies to fight many infectious organisms. They act as receptors, and some of them are hormones directly, like insulin, and some enzymes like trypsin, and even pigments like the hemoglobin found in blood. And this is very, very important. The most abundant protein in animal world is collagen, and the most abundant protein in the biosphere is rubisco, ribulose, bisphosphate carboxylase oxygenase, because it is the one which is found in plants. How do we understand and analyze the structure of a protein? There are different levels of proteins on the basis of structure. They are classified under four categories. The first one is the primary structure, the secondary structure, the tertiary structure, and the quaternary structure. The primary structure of an amino acid describes the sequence of amino acids. That is the positional information. Where does the an amine group actually is there in a free ionic state so that it can be bonded further, and where is the carboxylic acid that is present which can be bonded further. So the place where the amine group is there is called as the first amino acid or the N terminal amino acid, and towards the other end it is called as the last amino acid or the C terminal amino acid, and C representing the carboxyl group. Here one thing is very clear. We are coming into a kind of a nomenclatural analysis where we put the left end to a nitrogen and the right end to carboxylic acid group. The second is the secondary structure. Here we talk about one or more polypeptide chain that are folded together in the form of a helix. So we already know what is a helical structure. It is a rounded, folded, at the same time coiled structure. So here, one or more polypeptide chains, not only one protein, but two, I'm sorry, not only one chain, but two or three chains are combined together and they form an helical structure. And it will form only a right-handed helix. And examples include the keratin and the fibronin. Fibroin, I'm sorry, not fibronin, that is a separate product. And you can find here how it is arranged. You can find there is, when you take one strand of air, and inside the strand of air, if you magnify and see, you can find how this keratin is actually arranged. So this is the secondary structure of a protein. You can find the coil. At the same time, you can find the two coils being connected by the weak links, the saline and nitrogen bonds, and at the same time, the disulfide bonds as well. That is why we get a nature of the hair to be neither strong nor flexible nor weak. We cannot classify it because it comes under the secondary structural level of a protein. The third one is the fibroin, which is the silk protein. They contain the anti-parallel beta sheet, so it appears in this way. And here again, you can find there is an alloyside chain and a glycosyl side chain, which allow the complete close packing of the silk, which gives its strength to all kinds of sustainability. The tertiary structure of a protein describes the helical polypeptide chain from the secondary structure to be further folded like a hollow woolen ball. So as a result, we'll be having a three-dimensional view of the protein. And basically, this three-dimensional structure of a protein is very, very essential for many biological activities of the proteins. And this includes the biochemical reactions, the metabolic reactions, and so on. And finally, the quaternary structure. Here, more than one polypeptide chain is being engaged in formation of the structure, which means if one polypeptide chain is going to be there, which forms the primary, and then helical to form the secondary, and then a three-dimensional to form a tertiary. Here, more than one polypeptide chain will form the tertiary structure, and each of these will combine to form as a chain, and these are called as the subunits of the protein. So, for example, if you take hemoglobin, it will have four subunits, two alpha subunits and two beta subunits. So this sheet will give you a complete understanding of what is a primary protein and a secondary structure and a tertiary, and finally how it is assembled together form a quaternary structure. I'll pause here for 10 seconds so that you can have a glance at the picture. This is an extended version of the same picture. You can have a look at this as well. We'll go to the next biomacromolecule, the polysaccharides. The polysaccharides are polymers of sugars. That is the basic saccharides or the monosaccharides. Examples include starch, cellulose, glycogen, and inulin. Just to give a very small description, starch is basically a homopolymer of glucose. That is, this is one glucose, and this is other glucose, and this is the third glucose, and so on. When say so many glucose molecules are connected to each other, they form a polymer. And since they contain the same chain of molecules, it is called as a homopolymer. So starch is homopolymer of glucose, cellulose again a glucose molecule, but cellulose is found in basically the cell wall, while starch is a storage food material. Glycogen is again a homopolymer of glucose, but is found as a storage food material in case of animals, and inulin is homopolymer of fructose. And if you look at these polysaccharides, they are complex polysaccharides that is formed of amino sugars. That is, if you take chitin, it is a homopolymer of N-acetyl glucosamine. So they contain a sugar, and along with that, they also contain an amine group. And this particular chitin can be found in the exoskeleton of arthropods, for example, the crabs, the prawns, the lobsters. And apart from that, they are also found in the fungal cell wall. And we always know that chitin is a significant salient feature that is found in the cell wall of fungi. Just as we saw the formation of peptide bond in case of protein molecules, the polysaccharides also have a salient feature, the formation of glycosidic bonds. So the glycosidic bond is a bond that is formed when one monosaccharide is linked between two carbon atoms by the process of dehydration. So we can find one polysaccharide here and the other polysaccharide here, and both of them are combined together by the formation of a bond, which is the glycosidic linkage or the glycosidic bond, and in the process one water molecule is released. So this gives a complete diagrammatic picture of how the glycosidic bond is formed. I'll stop here for 10 seconds so that you can have a view at the picture. Starch forms a helical secondary structure. So the secondary structure is the one which forms the helical, which has deformed as we saw in the protein molecules, and it can hold the iodine molecules in the helical portion and hence can give a blue color. But cellulose has no complex helices and hence it cannot hold iodine. It is because of this we do the iodine test only to starch. So they form helical secondary structure, and because of this they can hold the iodine molecules. So from this nucleic acids we'll be seeing in the next video. So the next topic is about the nucleic acids, specific nucleic acids, that is the DNA and the RNA. Nucleic acids are heteropolymer of nucleotides. We already saw what are nucleotides. So they'll be containing a phosphate, a sugar molecule, and a nitrogen base. And if many nucleotides like this are going to be joined to one another, they form a structure that is called as the polynucleotide. The nucleic acids are basically of two types. One is DNA, deoxyribonucleic acid, and the other one is RNA, that is ribonucleic acid. And just as you can find, the deoxyribonucleic acid is a double-stranded helical structure, whereas the ribonucleic acid is just a single-stranded structure. The phenomenal conventional model that we know about the DNA is the secondary structure of DNA, that is the Watson and Crick double helical model. But there are many other models and dozen forms of DNA, that is aDNA, bDNA, cDNA, EZ, etc. So DNA basically consists of two polynucleotide strands and they are arranged anti-parallelly. So you can find they are anti-parallel to each other here, and they will be running as a double helix. That is, this is one helix and this is the other helix. If you take a DNA, and a nucleotide will consist of a nitrogenous base, a deoxyribose sugar, and a phosphate group. So the nitrogenous base and the phosphate group is going to be common in a nucleotide. But in DNA, what signifies that as a DNA is the presence of deoxyribose sugar. So if you take a DNA, the backbone of DNA is formed by the sugar phosphate sugar chain. So you can find there is a constant sugar and phosphate that is being bonded wherever I am circling. So there is a bond that is taking place between the phosphate and the sugar then and there, which forms the basic backbone. And there are steps that are formed in the middle with the help of the nitrogen pairs, so guanine, cytosine, thymine, adenine, cytosine, guanine, and so on. And so these nitrogen bases which form these steps in DNAs include adenine, guanine, thymine, and cytosine. So you can find that uracil is completely absent in these kinds of nucleic acids. And we should also know that the adenine always pairs only with thymine, that is A double bond T, where the 2 represents the 2 hydrogen bonds, and guanine always pairs only with cytosine by triple bond, that is by 3 hydrogen bonds. We can find that the phosphate molecule links the 3 carbon of the sugar, that is the freehand 3 carbon of the sugar of one nucleotide, to the 5th carbon of the sugar of the next nucleotide. So you can find this is going to be the 3 carbon and this is going to be the 5th carbon, and they both are linked together. The same applies for every sugar molecule. And between, I'm sorry, And between the phosphate and the OH of the sugar, that is between the phosphate linkage and the free hydroxyl end of the sugar, that is the alcoholic end, the third end, there is an ester bond that is formed, which is listed here. And that one ester bond on both the side is called as a phosphodiester bond. So the bond between the sugar and a nitrogenous base is called as a N-glycosidic bond, and the bond between the phosphate and the OH of a sugar is called as the ester bond. So there are two bonds which completely signify the stability of such a sugar, a nucleic acid molecule, that is a glycosidic bond and an ester bond. Let us take the structure of bDNA. The bDNA has totally 10 steps, that is, it has a 10 base pairs. And this bDNA is nothing but the Watson and Crick model of DNA. So you can find 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 steps totally. It is considered to be one full turn. And length of one full turn is going to be 34 angstrom. So how do you calculate the length of one full turn? The total distance between one major groove and one minor groove, that is one turn. So one major groove will be having a greater depth and minor groove will be having a lighter depth. And at each step of the strand, there will be a total turn of around 36 degrees. So every step will be turning 36 degrees. As a result of which one full complete turn will have 360 degrees. And if I'm going to tell there's going to be 34 angstrom for one full turn, then for one step it is going to be 3.4 angstrom.
This meeting is being recorded.
Next, we'll get into the process of metabolism. All the biochemical reactions that take place inside a living system is called as a metabolism. It includes all biochemical reactions, from food intake to food breakdown, creating energy, utilization of energy. All these things come under the process of metabolism. One example is removal of carbon dioxide from amino acids so that it can form an amine compound is also an example of metabolism. So when metabolism is happening, there are intermediate products that are formed in these metabolic reactions. So if you take, for example, glucose is giving rise to acetate. And in the process of glucose giving rise to acetate, there are products like trioses and pyruvates that is formed in the middle. These are called as metabolites. And these metabolites also should follow a specific a pathway, a metabolic pathway in which it can be degraded further or it is utilized for further processes. And they'll be having a definite rate and direction for this to happen. So it has got some specifications and it has got some methods in doing so. And the method by which it is done is called as the dynamic state. And as a result of this dynamic state and maintenance of equilibrium only, life exists. The metabolites are classified into two categories, the primary metabolites and the secondary metabolites. The primary metabolites are those which have identifiable functions. The functions are well in bigger notions and they can be identified. And they play a crucial role in physiological processes that are necessary for life. Amino acids, sugar, nucleic acids, lipids, vitamins all come under this category. The secondary metabolites are those which are not directly involved in growth, development, and reproduction, but they are indirectly involved. And they'll be exclusively found in plant, fungal, and microbial cells. And examples include alkaloids, toxins, lectins, drugs, etc. And this slide shows some of the important secondary metabolites that we come across in regular life, like the pigments, essential oils, the lectins, and drugs. And from where these are extracted, what are the examples of these secondary metabolites? I'll stop here for 10 seconds so that you can have a look at what are the things that is coming under different kinds of secondary metabolites. This is the continuation of the same. You can have a look at the slide as well. And same. So I'm just giving you the pictures of what are the different kinds of metabolites that we get from different kinds of organisms that we come across in daily life. So to keep it very simple, you can find the total number of metabolic reactions that is taking place in a human body. This is just a very imaginary hypothetical number and the scheme that is given here. And there are a lot of chemical reactions like which happen in our body. So there are series of linked multi-step chemical reactions where one product which is giving rise to another product will be the foundation for another product to develop, and then it goes on. So it is a multi-step chemical reaction. All these together will form the metabolism. The total metabolic pathway can be categorized into two. One is an anabolic pathway, and the other one is a catabolic pathway. Anabolic pathway means simpler molecules are formed from complex structures. So you can find the group of simpler molecules forming a complex molecules with consumption of energy. Catabolism means a big molecule is broken down into a simple molecule. That is, a complex molecule is broken down into a simple molecule with the release of energy. So this releases energy, whereas anabolism consumes energy. Let us see the examples. Acetic acid to cholesterol, amino acids to protein, and process of photosynthesis or anabolic processes. Conversion of glucose to lactic acid in the process of glycolysis, respiration are examples of catabolic processes. The release of energy through catabolism is stored in the form of chemical bonds. So it is not just stored in some other form, but it's stored in the form of chemical bonds. So whenever the stored energy is needed, this bond energy is utilized for all the works that is taking place inside the cell. One of the most important forms of such energy and the famous and the viable sustainable form of such bond energy is adenosine triphosphate. And it is also referred to as the energy currency. The living state, as we said earlier, in organisms, the metabolites are present in different concentration. So all the metabolites are not present in same unique concentration, nor they are present in the same amount in all the living organisms. For example, if you take the blood concentration of glucose, in a normal person it is 4.2 to 6.1 millimole per liter. The concentration of hormones should be in nanograms per milliliter. So systems at equilibrium cannot perform work. So if it is going to be at a perfect state, it is going to be at the constant level, it cannot, perform work. So the living organisms will work continuously so that they cannot reach equilibrium. So the living state is a non-equilibrium steady state with which it can perform work. So we can very simply tell the living process is a phenomenon by which we try keeping our body not to fall into equilibrium. So you prevent falling into equilibrium. And this is achieved only by energy input obtained from metabolism. So if you want to put your body under constant effort to prevent falling into equilibrium, you will have to need some energy, and that is the energy we get from metabolism. So no living state is without metabolism. It's a very important statement. We should keep it in our mind. Next, we'll go into the concept of enzymes. Enzymes are the biological catalysts. So catalysts are the substances which basically speed up or slow down a chemical reaction without actually getting involved in the reaction. So as it says, they influence the speed of biochemical reactions. And this statement is very important. All enzymes are proteins, but all proteins are not enzymes. Enzymes are specific. That is, each enzyme has its own substrate, I'm sorry. So it is just like the disease and antibody interaction. There are special things called as ribozymes. These molecules are nucleic acids, RNA, that behave like enzymes. Since they are ribose nucleic acids, and act as enzymes, they are called as the ribozymes. Enzymes form tertiary structure with some devices, and they are called as the active site into which the substrate fits. So, which means this is going to be the substrate, and if this is going to be the enzyme, there is a particular area where this enzyme will be exactly fixing inside a substrate, and that place is called as the active site. And it is only because of this active site mechanism that the enzyme can become functionable. This is also called as a lock and key model. Chemical reactions of the enzymes. The chemical compounds can go, undergo two types of changes. One is a physical change, and the other one is a chemical change. In a physical change, there is a change in shape or state of matter without the breaking of bonds. For example, the ice melts or the water becoming water vapor. But if you take in chemical change, which is also called as a chemical reaction, the bonds are broken and new bonds are formed. So it can be an organic or an inorganic reaction. So if you take an inorganic reaction, barium hydroxide plus hydrogen sulfate, sulfuric acid, it gives barium sulfate as salt with the release of water molecule. Here there is no active participation of carbon. So it's an inorganic reaction. If you take an organic reaction, starch is along with water will give a glucose molecule. So whenever this kind of reactions happen, the rate of physical or a chemical process or a chemical change or a reaction is the amount of product formed per unit time. You call it as alpha P divided by alpha T. The rate is called as the velocity if the direction is specified when a reaction is taking place. So just like the speed and the velocity. Speed with direction you call as velocity. In the same way, if the rate of a chemical reaction is specified in along with the direction, then you call it as velocity. And the rates of the physical and chemical processes are influenced by many factors such as temperature. And what happens is the rate of this usually doubles or decreases by half for every 10 degrees Celsius change in either direction. So this is something which has to be noted. So the rate will be doubling or it will decrease by half of the what is given in the parametrics if there is going to be a 10 degrees Celsius change in both the directions. And the rate of enzyme catalyzed reactions is very high, which means the carbonic anhydrase is the fastest enzyme. So if you are going to use an enzyme to catalyze a reaction, the rate is going to influence for a very higher rate. So if this is the basic reaction reaction, carbon dioxide plus water gives carbonic acid H2CO3. What happens is when we use this enzyme called carbonic anhydrase for this acceleration of this reaction, the reaction gets manifested 10 million times, which means in the absence of enzyme, only 200 molecules of carbonic acid are formed in half an hour. But in the presence of, in an hour actually, but in the presence of carbonic anhydrase 600,000 molecules are formed 1 second. So you can find the enzymatic action. In a metabolic pathway, each step is catalyzed by different types of enzymes. Here you can find in the process of glycolysis, that is, glucose is broken down to give pyruvate. And there are a lot of metabolites that is formed in the middle. You can find glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, and so on. And each of this conversion involves an active enzyme that is coming in the middle. And it is these enzymes which makes it very, very significant and breaking down and finally giving pyruvate. What is the nature of this enzymatic action or the catalytic action? The enzyme acts along with the substrate, and that is like a lock and key model action, and it includes the following steps. The first one is the substrate will bind to the active site of the enzyme. So you can find that there is a substrate with an active site which the enzyme can bind. And the enzyme and the substrate will go and bind along with the enzyme, and it forms an E plus X complex, that is the enzyme substrate complex. So this induces some changes in the enzyme so that the active site is tightly bounded to the enzyme and they form this ES complex or the enzyme substrate complex. This active site breaks the chemical bonds of the substrate. So what happens is at this active place, the substrate's chemical bond is completely broken and this form an enzyme product complex. And this enzyme finally releases the products and the free enzyme is also released out, and so that the enzyme can go and bind with the next substrate. So this pathway goes through some unstable transition state structures. So throughout the process, you can find some lot of intermediate structures that is also formed. Now, the concept of activating an enzyme or the process of activation energy. How do these enzymes speed up a chemical reaction? We know that the enzyme speeds up, as we saw in carbonic anhydrase. But how does this process actually happen? So, at this point, we need to know what is an activation energy. Activation energy is the additional energy that is required to start a chemical reaction. There is already a lot of energy, but apart from that, you need some more energy to kickstart a chemical reaction. That is called as the activation energy. If you take in case of an exothermic or an endothermic reaction, the substrate should go to a higher energy state, only then the reaction can happen. And this energy state is called as the transition state energy. So the activation energy is the difference between the average energy of the substrates and the transition state energy. So if the product P is at a lower energy level than the substrate, which you can find here, the reaction, if it is an exothermic spontaneous reaction, it requires no energy to form the product because it is already below it. And in a biochemical reactions, enzymes lower the activation energy. So the speed of the reaction increases. So when energy, activation energy goes less, the speed goes up. So what are the factors affecting the enzymatic action? The first one is temperature and pH, the concentration of the substrate. So as the temperature increases, the enzymatic reactions also go up. The concentration, and finally the presence of an inhibitor. Temperature and pH.
Enzymes show the highest activity at optimum temperature and pH. Why? Because they are proteins. So if you are going to keep the temperature high, the proteins are going to degrade. And if it's going to be low, enzymes become inactive. So activity declines below and above optimum level, as you can find from the pictures given in the graphical representations. At lower temperature, the enzyme temporarily becomes inactive. And at high temperature, the enzymes get destroyed because the proteins get denatured by heat. There are some inorganic catalysts which will work at high temperature and pressure. But even then, the enzymes will get damaged at high temperature, that is even more than 40 degrees Celsius. There are thermophilic organisms which have enzymes which are stable at even high temperature up to 80 and 90 degrees Celsius. And it is a result of which they got the name thermophilic, heat-loving organisms. The second is the concentration of the substrate. With the increase in substrate concentration, the velocity of an enzyme action rises at first. So when we increase the substrate concentration, the velocity, the rate of reaction will be increased and it will reach a very maximum velocity at one particular point. But this is not exceeded by further rise in concentration because the enzyme molecules are fewer than the substrate molecules. So once when all the enzyme molecules are bonded to the substrate molecules, there will be no free enzyme to bond with the new substrate molecules. As a result, the Vmax will not reach further state. Third one, the presence of inhibitor. The binding of specific chemicals shuts off the enzyme activity, which means if any other substance is going to come and form a substrate complex apart from the enzyme, then it is called as an inhibitor, and this process is called as inhibition. The inhibitor will closely looks very similar to the substrate, and it is called as the competitive inhibitor. So this will compete with the substrate for binding site of the enzyme. So even before the enzyme could come and attach, these inhibitors will form an inhibitor enzyme complex. So as a result, the substrate cannot bind and the enzyme action declines. There is an enzyme, a substrate, an inhibitor, and together they form a complex. This is non-competitive inhibition. But if the enzyme is going to, the inhibitor is going to occupy the place where the enzyme is going to come and occupy, then it is a competitive inhibition. One such example of a competitive inhibition is a malonate, which is similar to the substrate succinate. So it inhibits the succinic dehydrogenase in the following reaction: succinate to fumarate. But one advantage of these inhibitors is some competitive inhibitors are used to control many bacterial pathogens. How are the enzymes classified? The enzymes are classified on the basis of what functions they perform on the binding areas or the binding molecules. Oxidoreductases, dehydrogenases, transferases, hydrolases, lyases, isomerases, and ligases. Oxidoreductases and dehydrogenases are those which catalyze an oxidoreduction reaction between two substrates. Transferases will transfer a group from one substrate to another, and this happens by a very minimal catalysis. All can be catalyzed, but except the hydrogen group. Hydrolases are the group of enzymes which will catalyze hydrolysis of ester, ether, peptide, glycosidic C or P and N bonds, which means they are basically capable of breaking the bonds by using water molecules. Lyases are the group of enzymes which will help in removal of groups by mechanisms, all mechanisms other than using hydrolysis, and they will leave the double bonds also. So basically they will not break the unsaturated compounds. Isomerases are the ones which can catalyze the interconversion of optical, geometric and positional isomers. So basically they are capable of interchanging the structural models of different compounds. The ligases catalyze the linking of two compounds together. So they are capable of sticking just as the DNA ligase. So enzymes which catalyzed many bondings, they are called as ligases. When we are talking about enzymes, there is a concept called as the cofactors. These cofactors are non-protein constituents, so they are not enzymes, but they are bound to the enzyme to make the enzyme catalytically active. So if an enzyme has to be catalytically active, it is bonded to a non-protein constituent, and that is called as a cofactor. Apoenzyme is the protein part of the enzyme, and the non-protein part of the enzyme is called as the cofactor. So a cofactor plus an apoenzyme will be an alloenzyme, that is an active enzyme. But when the cofactor is removed from the enzyme, the catalytic activity of an enzyme is completely lost. What are the types of cofactors? First one is the prosthetic group, second one coenzymes, and third one metal ions. We'll take a 10-second break here. The cofactors or the prosthetic group are the cofactors which are organic, and they'll be completely bound to the apoenzymes. For example, the aim in the peroxidase and catalase. The second are the coenzymes, which are organic, and they have a transient binding to the apoenzyme, which means they will not bind completely. They can be easily broken, but still they have a binding with the apoenzymes. Many coenzymes contain vitamins, and all these vitamins are basically the coenzymes. And finally, the metal ions. They form coordination bonds with the side chains. So they do not form a direct bond, but they'll form a coordination bond that is very similar to an electrovalent bond. Not direct ionic, but something similar to that. With the side chains at active site. So at the site where there is actual, there is going to be actual binding of enzyme substrate complex taking place. These go and form a coordination bond there. And zinc is one such example, which is a cofactor of carboxy peptidase.